So this is our presentation, my presentation to you on the remainder of chapter 22 and chapter 23 as well. And so our goal is to try to understand those concepts. And if you have a question as we go through, please raise your hand. So we were talking about vestigial structures yesterday. Can anybody define for me a vestigial structure? Yes, Hayden.
Okay, isn't that when there's certain parts like hams in different species that have similar functions?
Okay, probably describing homologous structures.
Oh, yes, you're right.
Samantha?
Structures with little or no use.
Good, that's right, excellent. So when we talk about the bird's wings shown in this picture, these wings are not very useful for flight anymore. And so they may use these wings for possibly swimming or diving to obtain prey. Here we have a type of mole. It's actually got eyes that don't work anymore. And so these are vestigial eyes. These creatures, of course, spend time underground, so they really don't have much use for eyes anyway. So that's why nature actually permits the degeneration of the eye over time. So you could refer to those as vestigial eyes. The appendix was once thought to be a vestigial organ, but now we understand that it has more of an immune function now. They believe that it's involved in the repopulation of your natural bacterial population in your gut when that's needed. In this whale here, there are some bones in this rearward area where organs of the reproductive system attach, and so it has a very important role, but for some reason scientists are calling it vestigial, even though it has a very important purpose. But the reason they like to call it vestigial is because they like to think that somehow whales descended from a terrestrial mammal ancestor and lost its legs, and so this must be, it has to be the remnants of a pelvis from some terrestrial ancestor. Of course, if there was such an ancestor, we don't know really what it looked like, so the fossil record is disturbingly silent on that matter. But anyway, it does serve a purpose. So what counts as a vestigial organ to some people may not necessarily actually be a vestigial organ, so it's a little bit of a gray area sometimes. All right, so we talked about homologies. There are morphological homologies, which is when there are structural similarities, even if those structures might serve a different role, like, for example, the wing of a bird and the arm of a human. Certainly there are some interesting similarities. But there are also genetic similarities between organisms. And wouldn't that make sense? I mean, isn't that what we expect? If there's blood in all of these animals here, and that blood has hemoglobin, and hemoglobin carries oxygen, wouldn't we expect for that gene, for hemoglobin, to be present in all these different species? It seems reasonable to think that. Of course, there are some variations in the genes that determine hemoglobin production, and those variations are evident here. So there seems to be a lot more similarity between humans and this species of monkey here in terms of the genes that determine hemoglobin production compared to the types of genes that code for hemoglobin in the other species. So scientists will use this data to help basically explain their worldview on common ancestry among all organisms. All right? And you heard me right. The author of your book will say that a chicken is your cousin. That's not what Mr. Zimmerman's saying, but that is a point of view that's shared by probably 98% of biologists out there. Now, whether you think it's reasonable or not, you're going to have to come up with your own rationale on that.
What do you think? Do you think we come from chickens?
Well, because I'm a public high school teacher, I'm not going to stand up here and spend time going over my personal worldview.
It's like a yes or no.
I'll say this. I'm highly skeptical of any idea that is not well grounded on scientific evidence. That's the way I'll put it. But skepticism is a healthy way of looking at this. Anytime you want to think as a scientist, skepticism just means you're asking legitimate questions to try to understand the patterns we're seeing. So I know I didn't say yes or no, but I hope that helps you. All right, so looky here. this is a diagram that is supposed to show us the degree of similarity between organisms. Now, whether you want to interpret this as indicating degrees of relationship, that's up to you, but I think it's at least useful for kind of mapping out similarities. So for example, here we have an ostrich and we have a hawk. Both of those creatures have feathers, and so this feature here, feathers, is basically a defining characteristic of them. But you'll notice that feathers does not appear back here, because if it did, then our crocodiles would have feathers too, but they don't, obviously. So that's why this marker is located at this point. Notice that the amnion, which is basically a covering or the surrounding part of an egg, is present in all of these different species, whether they be mammals, reptiles, birds. However, amphibians lay eggs in water typically, and those eggs then become fertilized by the male, and so those eggs are not actually surrounded by an amniotic sac or an amnion barrier. So the amnion characteristic appears here, but it doesn't appear here because, after all, amphibians don't have the amnion. So that's how you read this. All right, any questions about that? Okay, convergent evolution. This is when, in some cases, it at least appears that two different species develop a variation that's similar. So for example, in South America, North America, Australia, there are species of squirrels that can glide. So they have flaps of skin connecting their shoulders to their hind limbs, and when they spread out, it serves as a way to prevent a catastrophic fall. This is an obvious advantage. You can glide from one tree to the other without having to get onto the ground and expose yourself to predators if you're one of these guys. So anyway, this is a physical feature that's found across several different species, and so an evolutionist may say that this independently developed over time, but because of similar environmental constraints, these organisms look similar. So they would call this convergent evolution. We should also consider the possibility that these two different species may have been distinct as far back as we can go, but that's not the way that this story is written here. All right? There are other examples that are alleged to be evidences of convergent evolution. For example, an evolutionist may say that the wings of a bird and the wings of a honeybee came about through convergent evolution, meaning that those wings are used for flight, but of course the structural differences are obvious. But if there's an environmental constraint that causes organisms to develop the ability to fly, at least that's the way the story goes, then you get that result. But is that really rational? Is that a reasonable point of view? You're going to have to decide that for yourself. Because if a feathered wing is to develop, there are a lot of genes that would have to mutate in a coordinated way at the right time, and to make or to ask all those right mutations to happen simultaneously is a statistical absurdity, at least seen from many people's point of view. So it's a difficult mathematical problem more than anything as to how those complex structures could have arisen. Biogeography. This has to do with the patterns of evolution that occur across our globe on the different continents, and how the geographic distribution of species might explain their physical traits. So species in nearby geographic areas resemble each other. Continental drift. All right. Some of you guys may have taken like an earth science class, maybe in middle school years. Do you guys know what Pangea is or was? Yes, Satori, go ahead.
So Pangea is the theory that all the continents were once combined together as like one huge massive land.
That's right. Yeah, so you can think of it as like a supercontinent. There's quite a bit of evidence that at one time such a supercontinent existed. So when you look closely at a map of the world, South America and Africa really look as though at one time they were joined together. They almost seem to fit together like a hand and glove if you were to cut out the pieces from a globe, a physical globe, and then piece them together. So, Pangea, what does this have to do with evolution? Well, in the time when Pangea existed, animals and plants could freely move about, and when they move about, they can carry genes with them because they have genes, right? And so when genes move from one place to another, you've got a lot of gene flow, and diversity has a chance to persist. Whereas if you're in an isolated place, diversity has a much more difficult time taking root. So conversely, if you think of like an island, on an island you may have a species there that is unique to any other place in the world because the genes that define that species are not free to move to other places because there's this huge barrier, ocean barrier. So whether or not a species is separated from other species, or whether that species can move to other locations where other species are, is a determining factor as to whether we'll get new species or not. Because speciation often happens when one species breeds with another. For example, if a coyote breeds with a wolf, you get a hybrid. And this is actually happening. Have you guys heard about this? None of you have heard about this. Yeah, it's happening in eastern Canada, mainly. And some of these coyote-wolf hybrids are starting to trickle into New England. At least this is what I hear in upstate New York. And so this is an example of how, because there's no significant physical barrier between wolves and coyotes, they are able to interbreed together, and then you get a new species. I don't think that the hybrid has a specific species name at this point, but at some point we could give it a species name, I suppose.
Aren't wolves like
Yep.
way bigger than coyotes?
Yeah, they are.
Like
You're right.
gigantic? Like compared
Yeah.
to coyote? Oh.
You're right. So in most cases, wolves are probably more likely to attack or feed on at least the young of coyote pups. But obviously that hasn't always happened. Sometimes coyotes and wolves will favorably interact with each other to breed. All right, so there we go. An example of an endemic species. We talked about Galapagos Islands. There are all kinds of species that live there that don't live anywhere else, like the gigantic tortoise. And there are several species of iguana that live there as well that don't live anywhere else. So how does speciation happen, at least in an island scenario? So let's say we have an island, and then there's this huge volcanic eruption. The island splits in half. There's an earthquake. Okay, and so many decades or centuries later, species A, which was split apart geographically, may begin, as they reproduce, to produce new varieties. So this would be species B. And then let's say there's another earthquake a few millennia later, a few centuries later, and then the island splits again. So species B may give rise to species C, and so on. And so whether or not these species are mobile and can swim across has a big impact on gene flow and on evolution. So these are examples of unique endemic species in Galapagos. Here are some various varieties of tortoises on the Galapagos Islands. And Darwin was known for studying these so closely. He knew exactly just by looking at a tortoise. If it was a sketch of a tortoise, he would know right away what island that species was endemic for. All right. So we've got 3 domains of life, 3 domains that you're responsible to know. There's the eukarya domain, which includes plants, animals, fungi. So this includes molds and yeasts and mushrooms and so on. It also includes protists, and protists are, a lot of them are single-celled organisms. They live in freshwater environments and in the ocean. So anything that has a nucleus in its cells is a eukaryote. So pretty big domain. Then there are 2 other domains, both of whom include prokaryotes. So we're familiar with bacteria. There are good kinds and there are bad kinds. When I say bad, I mean some of them are disease-causing for humans, and others are very helpful to us, like the ones that live in our gut, or maybe the ones that live in the soil and make nitrogenous nutrients available for plants to grow. And then there are prokaryotes. We could say that they're bacteria, but they're very, very special bacteria because they can live in very, very hot environments or very, very salty environments, or environments that are really toxic to most other forms of life. So for example, in the deep water ocean, there are hot springs that basically release sulfuric compounds into the ocean, and it's really hot water coming up from the crust under the crust. These archaea bacteria can survive. They can survive in that really hot water, and they can reproduce, and they're perfectly fine with it. Yellowstone National Park has a lot of hot springs, really hot water because of the volcanic activity deep underground. And so archaea will thrive there. How many of you have been to Yellowstone National Park in Wyoming? Anybody? Cool. What'd you think, Sydney?
It was really pretty.
Yeah? What'd you like most about it?
Probably just the fact that I got to see a bunch of buffalo.
Yeah, isn't it crazy? They're walking around all over the place, and you have to stop your car and wait for these herds to move across. And then the tourists are so preoccupied with taking pictures that you have to make sure you don't get in a car accident. But I was 15 years old when I first visited there, and this was literally like a month or two before the reintroduction of the wolf to Yellowstone, so I missed it by like a month. Does anybody know why they reintroduced the wolf to Yellowstone? Yes, Tori?
Because the like elk population was eating all the vegetation, so they were overpopulating like the whole
That's right.
park, and yeah, it was not a good time.
Right, yeah. So in 1988, so I was 8 years old, there was a horrible fire, forest fire in Yellowstone, and so the forest was having a hard time rebounding. Trees were not really growing that well because elk were coming along and gobbling up all the seedlings. But by bringing the wolf in, we were able to put a limit on herbivore populations, and then plants were able to grow better. It's a little off topic, but I suppose it does link to evolution to some extent. All right, I'll show that later. What I'm going to do now is we're going to look at chapter 23. So let's turn there for a second, please. All right, Tristan, you ready? Here we go. So on the AP exam, there are there's probably going to be at least one question about the frequency of alleles. Can anybody refresh my memory on what an allele is? What's an allele? Things that code what? Nothing? Oh. Yes. True statement. Alleles do code for physical traits. But what is a difference between a gene and an allele? Yes, ma'am.
An allele is the different variations of a gene.
Excellent. An allele is a variation of a gene. So we're talking about really specific forms of genes when we talk about alleles. So we're going to learn how mutations in sexual reproduction each produce genetic variation. We're going to learn about a theoretical state of a population called the Hardy-Weinberg equilibrium. Basically, it just states that, okay, if a population is to not evolve, then what are the conditions that need to be in place if that population is going to stay the same? And that's what Hardy-Weinberg is, and I'll talk about that later. We're going to learn how to calculate the frequency with which certain alleles may occur in a population. So we're going to be doing a little bit of math. Hopefully you guys have your calculators. All right, so micro means small, right? So when we talk about microevolution, we're talking about subtle changes that occur in a population. So for example, the domesticated dogs that we observe descended from a canine ancestor that most likely was a wolf of some kind. So we've got a lot of different varieties. And so we would call this microevolution. Microevolution. Now, macroevolution is on a grander scale, and it's more like an idea or a theoretical construct, which involves bigger changes, if it did happen. So if one were to actually believe that humans are related to insects through a common ancestor, there'd be a lot of changes that would have to happen if such a transformation actually did happen. So that idea is what we call macroevolution. So I've talked about Charles Darwin. In a previous screencast, I mentioned that Charles Darwin lived at the same time as Gregor Mendel. Gregor Mendel was doing genetic experiments with pea plants, and Charles Darwin was formulating his theory of evolution by natural selection. But because Darwin hadn't actually published until 1859, and Mendel was still doing his experiments, and I don't know if he had published around that time or a little bit later. I'd have to check my stats on that. But these guys didn't really know about each other. It would have been nice if they knew about each other because then they could possibly inform each other's work. But both of them were working on this idea of genetic variation. How do you get genetic variation? And how do the factors that determine traits get passed on? And how do they get manifested in later generations? So, there are different kinds of mutations that do have a role on evolution. Point mutations. These are changes to just one base pair. So you might remember adenine, guanine, cytosine, thymine, and so on. So if there's a change to that base, if maybe a T is substituted with A, that's a point substitution. Chromosomal mutations, on the other hand, have or will experience either a deletion or a duplication. Cool. Just checking to make sure my clock is running there. All right. Sexual recombination. There are several ways that genetic information can be sexually recombined. We already learned about them. First, crossing over, which happens during meiosis. Second, independent assortment of chromosomes during meiosis. There's a third one. Here we go. Also, random fertilization. It's very rare that random fertilization happens. It may happen from time to time, but with mammals, for example, mammals tend to be very picky about who they're going to mate with. Usually. Not always. Maybe they should be more picky sometimes, but okay. Population genetics. Study of how populations change genetically over time. That's a discipline that's really important for any conservation biologist, any ecologist. How do we define a population? Well, it's made up of organisms that are usually of the same species, and they have the potential to interbreed. Most likely they live in the same area, and they're going to produce offspring. Sometimes the word population and species are used interchangeably. Sometimes. Okay. A gene pool. What is that? Well, a gene pool is basically the collection of genes for a population. So, for example, for a human population, if we consider all the genes that are in a human population, we'll refer to that as the gene pool. Or if we consider the coyote population, especially here in the valley, and all the genes that make the coyotes what they are, that's referred to as the gene pool. Now, if an allele is fixed, here's what that means. That means that that allele is present in most or all of the individuals in that population. It's probably an allele that determines a favorable trait. Otherwise, it wouldn't be present as much as it is. Sometimes that's what happens. Or sometimes if a population gets really, really, really small because it's becoming an endangered species, now there's a lot of inbreeding going on, which means that alleles that are the same are getting combined with more frequency. And so then all the other variations of genes are being eliminated. So sometimes alleles get fixed because the population is under tremendous stress and it's becoming very, very small. So the more fixed alleles in a population, the more fixed alleles a population has, the lower the species diversity. So it's usually not considered a good thing when only one allele for a particular character is present. Usually we want more alleles because we want diversity for a healthy population. All right, so Hardy-Weinberg, equilibrium. So this was named after a couple scientists that were really interested in mathematics and how it applies to genetics and biology. So the allele and genotype frequencies of a population will remain constant from generation to generation. This is the principle, all right? With me, Riley?
Yes.
Okay. So if there's not going to be any changes happening, what kind of conditions need to be in place? That's what we need to think about. When we say a population, or when we say the alleles in a population are in equilibrium, we just mean that there's not any change. There's not any change in the frequency of those alleles. So, if a population is not evolving, here are the conditions that need to be in place. First, no mutations allowed. None. No mutations allowed. Is that realistic? No, it's not realistic at all. So this is kind of like an imaginary scenario. It's a hypothetical. Random mating. For most populations, that's probably not that realistic either. But let's imagine that such a thing could happen. No natural selection. Well, I mean, we learned yesterday that humans, to some extent, may be buffered from natural selection because of the healthcare advances that we're using to keep people alive that otherwise would die, which obviously it's a good thing if we can save those individuals' lives. But other species, but besides humans, other species cannot afford that luxury, or they don't have that luxury, I should say. So natural selection is normally at work. So this is also somewhat unrealistic for most populations. Extremely large. Hey, I can think of a lot of species that are endangered, and their populations are getting pretty small. So again, this is not realistic for many populations. No gene flow. That means there's no migration allowed. There's no immigration, emigration. All right. So organisms are not really moving around too much at all. Is that realistic? Well, in most scenarios, that's not realistic. Organisms tend to move if they can. If they have to find resources elsewhere, they're going to get out of town and find it, if they can. So what's the point? If all of this, if all these characteristics of a population are rare or impossible, then why are we even considering that all 5 of these conditions could ever be met? Any ideas? So remember, if a population has these characteristics, there's no evolution going on. So this actually serves as a baseline off which to measure evolution. So if a population is changing, we can ask the question, is it more because there is non-random mating, or is it because natural selection is exerting pressure, or is it because there's maybe a really small population or a large population? Are there genes moving around? So we can ask these kinds of questions when we observe that a population is changing over time. All right, ready for math? Hope you've got your math brains. Your mathematical gray matter will be oozing out of your ears by the time we're done with this. But it'll be fun, I promise. You're going to have smiles on your face because you'll walk out of here knowing you learned something. Yes, if you want to get out a calculator or use the calculator app in your phone, you certainly can. Let's represent 2 alleles as P. Let me backtrack. The frequency of one allele may be designated as P, and the frequency of the other allele would be designated as Q. So, for example, let's consider capital A and little a. So, the frequency of capital A is designated with this variable, P. The frequency with which the little a allele occurs in a population is designated with this algebraic expression, or this variable. So these are frequency variables, all right? So for example, the frequency of capital A and the frequency of little a, actually I should write it this way, will always equal 1. It's written right here. Okay. So 1 is the totality of the genes for that particular character. So let's say this is for earlobe attachment. All right. So we learned that a dominant allele determines free earlobes, and the recessive allele determines attached earlobes. But the capital A is the dominant allele, correct? All right. So the frequency with which this allele occurs plus the frequency with which this allele occurs equals 1.0, or 100%, of the gene pool for that particular character. All right, so 1 means the totality of the gene pool. All right, so now let's consider not the gene pool, but let's consider individuals. By the way, I should backtrack for a second. What do I mean by gene pool? If this circle represents the gene pool, and I have a bunch of capital A's and little a's available to be combined in the gene pool, remember that if a little a combines with a capital A, we'll get a capital A, little a combination in an individual. So when you see these letters, don't think of them as representing individuals necessarily. Think of them as like sex cells that have the potential to combine. All right, so here's sperm cell, egg cell, All right, so when individuals reproduce, those sex cells are combining. All right, now, and I should explain that when you imagine the gene pool that I just showed you earlier, you need to think of this formula, P plus Q equals 1. All right, so this formula is supposed to show us that the frequency with which individuals that are homozygous dominant occur, plus the frequency with which heterozygotes occur, plus the frequency with which homozygous recessive individuals occur, will always equal 1.0. In other words, 100% of the individuals in that population in regards to this character. Any questions so far? So p squared is just means p times itself, right? All right, now we're going to think of an example of how to apply this. Let's say we have an allele for red and an allele for white. And when these combine to make a flowering plant, we get pink offspring. All right? Now, if the alleles for white combine, we'll get white offspring, and if the alleles for red combine, we'll get red offspring. Make sense so far? Because this is incomplete dominance. We learned this last unit. Now, let's say that in a gene pool, the frequency with which the R allele occurs is 80%, and the frequency with which the W allele occurs is 20%. And last time I checked, 80% plus 20% makes 100%. So in other words, students, 0.8 plus 0.2 equals 1.0. All right, so that's the total. That's the totality, 1.0. So if 80% of the gene pool is made up of the alleles for R, for red, I should say, 0.8 times 0.8 would be the frequency with which we would get a RR combination. In other words, that's the frequency with which we're going to have offspring that are red. We just multiply the chance of this being given times the chance of that being given in a sexually reproducing event. All right. What's the chance we get pink? Well, one parent could make or produce a white, I should say a sex cell with a white allele. The other parent could do the same. So 0.2 times 0.2 represents the chance that we'll get white offspring. What if this sex cell combines with this one? What's the chance of that? Multiply 0.2 times 0.8, you get 0.16. So when we combine the alleles together, we get offspring. And so the PQ is the chance that we get an RW. On the other hand, you could get WR, which is another 0.16. So if you add these together, if you add up PQ to PQ, I'll demonstrate. The chance of getting a combination like this plus the chance of getting a combination like this, which essentially is the same thing in terms of outcome, is the same thing as 2PQ. All right? So that is where the part of the formula shown earlier comes from. The 2PQ is the result of adding the two heterozygous possibilities together. All right? Cool. So moving on. Let's consider for a minute how this all worked out. The chance, so if we ask the question, what was the frequency again of having an allele that determines red pigment? You just add up all the individuals, or the percent for the individuals that are red, plus the percent for individuals that are pink, because after all, they do have the red allele, and you get back to this frequency for that red allele. All right, and then if you add up the frequency with which we get white plants, and add those to the individuals that are heterozygous, then you get, again, the frequency that white allele occurs in individuals in our population. All right, so here's number one. It says if you are given the genotype capital A, or capital A, or little a, you can calculate P and Q by simply adding up the total number of capital A and little a alleles. If you know the phenotypes if you know the phenotypes, then you can use little a to find Q squared. So if you know the frequency of, let's say, the little a, it's very easy to find out what Q squared is, and I'm going to demonstrate that for you in just a second. We're going to use this formula, p squared plus 2p cubed plus q squared, to find the genotype frequencies, the expected genotype frequencies. And if p and q are not constant from generation to generation, guess what? Population's evolving. Maybe one allele is becoming more and more frequent because nature is selecting it, and then Hardy-Weinberg principle no longer holds because there's some kind of selective pressure going on there. All right, here we go. This is where we're going to use our calculators. So let's say we have, I'm going to have you guys help me out here. I'm going to trust your calculating abilities. The frequency with which a homozygous dominant individual is occurring is, well, I should say the raw numbers are 1,469 for this. The number of individuals that are heterozygous is 138, and the number of individuals that are homozygous recessive is 5. So who wants to add up all three of these numbers? What are those?
1612.
1612?
Yeah.
Okay.
So
You want to check that for me, guys?
Yep,
It's on the
16,
board.
12.
Oh, it is? Okay. Oh, thank you. It is. I wish Mr. Zimmerman would pay attention. All right, good. So if you add those up, that's what we get. We're going to then consider what the allele frequencies are. Okay. I'm going to just pause this video here, and I'm going to give you guys a chance to kind of work your way through this, and I'll give you a little helping hand here and there. And then I'll restart this in a minute. That way our seniors can see our... All right, so here's what we do. Why do we take that number 1612 and multiply by 2? Well, here's the issue. Remember that an individual has two pairs, or I should say two alleles for each pair. So we're actually going to need to calculate the total number of alleles in the gene pool, which is double the number of individuals. Does that part make sense? So we double our number of individuals because each individual has two alleles for a given pair. Any questions about that? So we end up with this number 3,224. Okay. Now, what's the significance of 1469? Well, students, if we want to know the frequency of individuals that have the homozygous dominant genotype, this is what we do. We take 1469 and we divide, we multiply it by 2, and then we're going to get our, let me backtrack here. Okay, so the number of capital A alleles in a gene pool is given, okay, so we're actually not trying to find out the frequency of homozygous dominant individuals right now. We're just trying to figure out what the number of alleles, or capital A alleles, is. So we take this number, we multiply by 2, because each individual has two alleles for a pair. Then we add 138. Why do we do that? Because we have 138 individuals that have one dominant allele. Okay? Then what do we get? We're going to get this number right here, 3,076. What is this number here? That would be the number of dominant alleles in the gene pool.
So you have to divide that by 3,224, right?
Eventually, yes. Yep. So then, how do we find out the number of, or the total number of recessive alleles? Well, how many individuals have the recessive allele? Well, we have 5 here, but remember we also have to multiply that number by 2 because all these individuals have 2 alleles, right? So we multiply 5 by 2. And then we add 138. 138 individuals are heterozygous, so they have one copy of that allele. Now, why don't we multiply this times 2? Well, these individuals only have one copy of that recessive allele. So then we get 148 recessive alleles in this gene pool. 148 recessive alleles are in this gene pool. And Tristan kind of forecasted what our next step is, and I'm going to scroll a little bit here.
Yeah, this makes sense.
All right, cool. So let's take, where did we get this number? 3,076. Oh yeah, 3,076 was the number, the total number of dominant alleles in our gene pool. Remember my circle with the gene pool? Remember this? And I was like, okay, the gene pool consists of some dominant alleles, consists of recessive alleles, and so on. This is the gene pool. So how many dominant alleles are there in this gene pool? There are 3,076. To find out the frequency, in other words, the percentage of alleles in the gene pool that are dominant, we divide the raw number of dominant alleles by the total number of alleles, whether it's capital A or little a. And then we get our frequency. Check it out. So now we've got a little over 95% of the gene pool is made up of this dominant allele. Next, I'll scroll. All right. Next, where did the 148 come from? What's the significance of that? Well, remember, 148 is the number of recessive alleles, the raw number of recessive alleles in the gene pool. And so we take that number and we divide by the total number of alleles in the gene pool. And then we get our frequency with which that recessive allele occurs in the gene pool. All right, so students, if you add P and Q, what do you get? If you add them together, what do you get?
One.
1. There you go. So 1.0, that's the totality of, or I should say that represents the totality of alleles in that gene pool. So P plus Q is always going to equal 1. It has to. All right, so guys, what have we figured out so far? Let's just summarize what we've learned. We've learned when we're given raw numbers for certain genotypes, we can figure out the frequency with which a capital A occurs in that gene pool. We can also figure out the frequency with which a little a occurs in that gene pool, and that's what we've done here. Now, here's the easy part. This is the easy part. What if I want to know, all right, let me backtrack for a second. Let's say that this population is mating. They're randomly mating, all right? And if they're randomly mating, what can we expect in the next generation? What kind of frequencies would we get in the next generation for each of these genotypes? In other words, how many of these individuals are going to be capital A, capital A? That's what we need to think about. So here we go. To find out what the frequency of homozygous dominant individuals is, you simply take P and square it. So 0.95 squared gives us this.
Wait,
That means, yeah, go ahead,
where'd you
Tristan.
get the 0.95 from? Sorry.
Okay, good question. The 0.95 is the frequency with which a capital A occurs in the gene pool.
Okay, I did the 1,469 divided by the 1612, and I still got the 91%.
Oh, okay.
Down here for the frequency of the AA genotype. And I was wondering why your numbers were different, but that makes sense because it's basically the same way of solving it, or it's not the same. It's basically getting the same answer, but it's a different way of solving it.
Yep. So it makes sense now?
Yeah.
Cool. So you just square P, and that'll tell you, okay, 91% of the time, if there's random mating going on, we would expect that 91% of the offspring are going to be homozygous dominant. That's what that number means. Now, for this population, what if there's random mating? What's the chance or the frequency of getting homozygous recessive individuals? In other words, I should label these. This is the frequency of getting capital A, and this number represents the frequency of getting little a. So that would be 0.002%. It's a pretty small possibility, right? So 0.002, I said 0.002%, it's actually 0.002, which would be, you'd have to move the decimal to determine the percentage, of course. Yes,
Do you mind if I take a picture of the board?
you sure can. Yep. Yep. In fact, yeah. All right, 0.002 is the frequency with which we get a little a combination. How about 2pq? How do we do that? Okay, remember that 2pq is the formula for determining the frequency of getting a heterozygous individual. In other words, a capital A, little a combination. So how do we figure that? Let's just plug the numbers in. That's all you do. You plug this number in for p, plug this number in for q, multiply by 2. That means 8.7% of the time, we're going to get offspring that have a capital A, little a combination. Okay? So if you can figure out P, you can figure out everything else. You don't even need to figure out Q off the bat. If you already have a way of figuring out P, or vice versa, you can figure out all the rest. So, any questions about that? Cool. All right, so maybe it wasn't that bad. I kind of painted a grim picture that was going to be horribly difficult mathematics, but we're going to get more practice in the coming days. All right, cool. Let's go back to our presentation. So we just worked through this. So again, just in summary, if those frequencies don't change from generation to generation, then we can say that there's no evolution happening. And that's the Hardy-Weinberg principle. If evolution is happening, then those frequencies are going to change from generation to generation. All right. So if there are mutations, these are often rare, small changes, and they can cause changes in allele frequencies.
We don't.
Oh, you don't have this?
I don't think so.
Yeah, we do.
Yeah, you skipped this one.
Did I? Oh, okay.
What? I didn't even see this.
You skipped the Hardy triphosphate.
Oh.
Did I? Oh, well, yeah, I did. I skipped over that because we actually did that in class. I'll go back and show you.
So where is this?
You're talking about this slide right here, right?
Yeah.
Oh, yeah. I guess I did.
Where was the one we were just on?
It's
It's like 2 pages back.
How about, why don't we finish chapter 23 and then we'll come back to this slide? Because this is the one you were talking about that I skipped over, right?
yeah.
Okay, I'm sorry. Yeah, we'll come back to this a little bit later. All right, so if there's non-random mating, this will affect genotypes as well. So think of like birds. Female birds are very choosy about their mates. They'll tend to pick the male that's either better colored or has a nice song to listen to or puts on a better dance. What are some other traits, Satori?
Oh, I was just saying the dance.
Yeah, the dance is important oftentimes. It makes a difference between whether you can pass on your genes or not if you're a male.
Better
Yes.
Nest.
What's that?
Better nest.
True. In some cases, males can impress the females with the nests that they build. So, why do I mention this? Well, this is what they call sexual selection. And the genes that determine bright colors and pleasant song and all these other traits are going to become more abundant in a population if females continue to be very choosy about who they're mating with. And so this is an example of how non-random mating, which I just described, can result in changes in allele frequencies. Certain alleles may become more frequent as a result of that. Oops. Natural selection is a major cause of evolution, as we've just learned. There's something else called genetic drift and gene flow, which we're going to talk about right now. Genetic drift and gene flow. This is fairly straightforward, but it's important to understand. Nature selects individuals that are best suited to survive in their environment. And so if they can avoid predators, or if they're efficient at finding food, or if they can regulate their energy reserves better, these are advantageous characteristics. Nature will select based on that. All right. So some characteristics may actually shift or change depending on whether the environment is changing. Now, genetic drift is basically when a population is starting to lose its diversity. Small populations are really vulnerable to genetic drift. There's a greater chance that certain alleles will start to become less frequent and others will become more frequent during genetic drift. And there are two kinds of genetic drift. There's founder's effect and bottleneck effect. So if there's a population that becomes endangered, in other words, as numbers shrink to very small numbers, now that population is going to be inbreeding. Do you guys know what inbreeding means in a biological context? Inbreeding. Like, what's the result of inbreeding, I should say?
Bad things.
Bad things, like
Yeah.
what?
Like chromosomal defects or just genetic defects in general.
That's right.
Because they're they're genetically too similar to have variation.
Right. Yes. So the recessive alleles that often cause genetic disorders are combining with higher frequency in populations that are really small or in populations where closely related individuals are meeting together. And this is why in the state of Arizona and pretty much every other state as far as I know, it's against the law to marry a brother or a sister because the government's trying to basically control the pollution of the gene pool, essentially. What's that, Satori?
I said except in Alabama.
Yeah, well, I'm sure the law is broken from time to time in certain places. I lived in Pennsylvania before I came here, and the Amish were practically our neighbors. And not that they have brothers and sisters marrying, but there are first cousins that marry. It's very common. And they do have certain genetic diseases that are fairly frequent in their population, certain kinds of dwarfism, for example. So that's some degree of inbreeding that's going on there. So genetic drift is sometimes caused by inbreeding, or sometimes a random event like this could happen. For example, let's say I'm mowing the lawn, and let's say I stupidly had a glass of wine before I mowed the lawn, and I steer off track, and I go off into my neighbor's lawn, and I
One glass of wine did
yeah,
all
one
that?
glass of wine, yeah. Well, see, I'm very sensitive because I hardly ever drink wine, so anyway. But let's say I steer off track.
Bounce it off the
And the flowers get mowed over. Oh no, now I have to write an apology to my neighbor because I'm too ashamed to go tell him face to face. Let's say that just by chance, the white flowers are destroyed. Oh no, now we have red flowers left over. Now, in my state, I certainly wouldn't have purposely gone over only the white flowers and the pink ones and left the red. Maybe this was just random chance, mostly the red flowers were left over. Well, that's an example of genetic drift. Now, I know you guys are never going to forget genetic drift because I just gave you this wild scenario that never happened, and it was very imaginative, I know. But that's genetic drift.
the strongest glass of wine ever.
Yes.
But
Okay.
it's like aged over
Yeah.
500 years.
All right. So, what's founder's effect? You guys have maybe heard of polydactylism. It happens in cats and sometimes dogs. It also happens in people from time to time. There's a recessive allele that causes this. In Amish communities, this is a little bit more common because when the Amish ancestors moved to North America, there may have been a few individuals that had polydactylism, and so they were passing on these genes for polydactylism, and so that particular trait was more and more common. The frequency for that allele was very common in those populations. All right, so we're going to stop there, and we'll continue tomorrow.
This was a good lecture.
Oh, well, thank you. I appreciate it.
Time went by very fast.
Yeah, so, can you stay for just 1 second? I know I've been going through a lot of notes lately, but I knew because of the content and the fact that it's not quite as dense as what we've covered before, we could afford to do that. I'm kind of gearing you guys up for the mock exam on Saturday. Tomorrow we'll do some more practice with allele frequencies. So, by the way, what's due Friday? Yes, Active Reading Guide, Chapter 22. See you guys later. Thanks.
It's not a secret.